For the next 3 days, we'll be speaking to some of the leading experts on research, advocacy, and treatment of rare diseases. And we'll start the training off with Dr. Eric Green, the director of the National Human Genome Research Institute. Dr. Green, welcome to our fellowship program, and thank you for joining us.
Well, thank you, Rachel. Happy to be here.
So we thought it would be particularly appropriate to start with you as we unpack this issue over the next few days. And I would like to think that based on my research and based on the conversations that I've had with people like you, that perhaps in the world of research and treatment of rare diseases, we've reached an inflection point of sorts. So before you show us your presentation, tell me if I'm on the right track.
No, I think you very much are on the right track. We've seen developments over the last several decades, but in particular, even the last 10 years that have greatly accelerated our understanding of rare diseases and also put us on a trajectory to think about more powerful approaches for diagnosis and in some cases, potentially treatment. And all of this has, or much of this has been an outgrowth of rapid acceleration in genomics, the field of study that I'm responsible for at the National Institutes of Health. So I think you are on the right track, and I hope during my talk, I'll be able to provide the participants a really valuable context for a lot of the talks that are going to follow.
You can read more about Dr. Green's work, again, by clicking on the link to his bio at nationalpress.org. So, Dr. Green, the stage is yours. Please give us an overview of rare disease.
Absolutely. Thank you so much. So I'm delighted to be here. I'm certainly excited to be participating in this program. And I was assigned the title Rare Diseases 2020, an overview, but actually I think maybe the more accurate reflection of what I'm going to try to do for the next 30 minutes or so is to really show you how rare disease advances have built up because of an unfolding story of this relatively young field of genomics. And what this is going to do, I think, is very much set a context for many, if not all, of the talks that are going to follow over the next 3 days. So having me go first, I think, is really valuable. I'm going to sort of fly at a very high altitude to give you a broad context of where all of this exciting set of developments are growing up upon, but I think you're going to see that many of the other speakers are going to take you to a much lower altitude and give you a lot more details about some of the things I just may touch on. So it's not an accident that there's a lot of excitement about rare diseases currently in 2022. But what I will tell you is that much of this has been a result of an evolving story of human genomics. And I don't have time to describe to you all of the details of this story, but like a lot of stories, it unfolds in a series of chapters. And I'm just going to briefly introduce you to these chapters and show you along the way where rare diseases sort of came into play and contributed to this growing, rich story of this field of human genomics. Now, I admit I will also be telling the story very much through the lens of a research funder, since I direct one of the NIH, in the United States, the National Institutes of Health has 27 institutes and centers, and I direct the one that is focused on genomics. And so we think a lot programmatically about how to build the field and make scientific advances in our research area. And you will see the way I tell the story is very much through the perspective of somebody who oversees the funding of genomics research at the National Institutes of Health. I'm also telling the story as somebody who's lived the experience of human genomics from the beginning of the field. I've been involved in the field essentially since its inception. Now, like any good story, it starts with a prologue, and the prologue here is relevant. I mean, the science of DNA grew substantially throughout the last century. Particularly notable events included the discovery of DNA's double helical structure in the 1950s, followed by the elucidation of the genetic code in the 1960s. But in many ways, the most relevant part of the prologue associated with the story of human genomics was the molecular biology revolution of the 1970s and the 1980s. And the big question for this prologue was, can we develop the tools for comprehensively exploring genomes? This is going to turn out to be fundamentally important for rare disease research, as you will see. Well, the molecular biology revolution was specifically associated with the development of such tools. For example, those that could be used to clone DNA, manipulate DNA, that is recombinant DNA cloning, such as represented on the left. Later on, there was methods that came about for sequencing or reading DNA, reading out the 4 letters of the 4 different nucleotides that make up the sequence of DNA, shown in the middle. And then eventually even methods for selectively amplifying DNA through amplification methods such as that shown on the right. Now, as the tools of molecular biology got better and better throughout the 1980s, the notion of performing studies that would allow you to study all the DNA of an organism, and that's known as its genome, began to get serious consideration. And there was, of course, particular interest in comprehensively studying the human genome due to the compelling challenges in human and medical genetics that needed a better understanding about the structure and the function of the human genome and more powerful approaches for studying human DNA. And here, think about rare diseases, having a genetic cause, having the ability to comprehensively study a person's genome might provide the tools to allow us to better diagnose, eventually understand rare diseases, and maybe even one day be able to treat them more effectively. All right, I'll try to continue. I'll have to probably be much more abbreviated in my comments so you keep this program on time. So I think where I left off was this naming of this new field of genomics in 1987. And there was a deliberate reason why a new field was named and a new term was coined of genomics. And that was because in the late 1980s was this growing interest at the idea that we could embark on a large project to comprehensively, once and for all, characterize the human genome. And that leads to this first chapter of human genomics. An effort, of course, that effort was known as the Human Genome Project. And the Human Genome Project and this chapter really was about a single question: can we map and sequence the human genome? And it really was an audacious challenge ahead. Something was very unusual to try to pursue something like that on a very large scale. A series of strategic visions were published that guided the project from beginning to end. I was involved in the project from beginning to end. And over the next 13 years, this remarkable endeavor played out. And among its goals were determining the first sequence of the roughly 3 billion letters in the human genome, and all of human DNA represented by these 3 billion letters. So this was a remarkable success story, and because it provided the first real insights about the complete set of DNA instructions carried in every human. And I should point out that from a journalistic point of view, these are sort of very famous covers of major scientific journals, reporting, in this case, the first draft sequence of the human genome in 2001. And some of you might have noticed that those same 2 journals featured last year in 2021, the fact that we've now reached 20 years of having at least a draft sequence of the human genome in hand. And of course, next year will be 2023, and I will tell you that we'll be then celebrating the 20th anniversary of the completion of the Human Genome Project and the generation of that first high-quality sequence of the human genome. Now, the completion of the Human Genome Project really marked the end of one chapter in human genomics, but the immediate beginning of another. And this chapter 2 of the story of human genomics really saw a major expansion and maturation of the field. And really the key question for this chapter 2 in genomics is, can we use genomics to advance knowledge about health and disease? And now we can start thinking about disease and rare diseases as an example. And so our institute put out, the day the Human Genome Project ended, a strategic vision of what needed to be done in genomics beyond the Human Genome Project, with a particular focus on health applications of genomics. And you could see from this iconic graphic how the Human Genome Project was now going to provide a robust foundation on which we would build knowledge connecting genomics to biology and to health and to society. And that strategic vision really was very important for the idea of beginning to think about how we were going to want to make connections between genomics and medicine. Now, this was not a new idea. This was one of the key rationales from the beginning of the Human Genome Project. But with the completion of the Human Genome Project, and again, as journalists, you'd be interested to see that both the popular press, such as the New York Times Magazine, or the scientific press, such as Science Magazine, shortly around the time the Genome Project ended, began juxtaposing the two words genomics and medicine, leading to this phrase genomic medicine, recognizing that was going to be the next ambitious goal in the story of human genomics. I will tell you that when that Genome Project ended, we could say the phrase genomic medicine, but it was certainly not in focus yet. We sort of generally understood what we might be talking about, but we didn't really know what that was going to look like operationally. We could define it, and we did. And it was sort of an emerging medical discipline that involves using genomic information about individual patients, recognizing that every patient has a slightly different genome. Could you use knowledge about differences in each of our genomes to better tailor our medical care and to better manage our health? But of course, this was not going to be simple. The starting line for this journey, the next stage of genomics, was going to be the Human Genome Project's completion. That was the starting line, and the finish line was going to be eventually realizing genomic medicine and changing the practice of medicine. And just like the Genome Project, it would involve a lot of steps, and we should be prepared to recognize we won't know what all of those steps are. And we will have to figure it out as we go along, and slowly but surely, just as with the Human Genome Project, check these steps off as we accomplish them. Also, like the Genome Project, it was not going to be a sprint to realize genomic medicine. It was going to be much more analogous to a marathon, a long run, many people, many disciplines, many funders, many countries, all contributing to the building knowledge about the human genome based on the product of the Human Genome Project as we inched our way closer and closer to seeing genomic medicine become a reality. But each of those steps reflected a barrier that needed to be knocked down. And the very first barrier that was very clear was going to be needed to be knocked down after the Human Genome Project was the cost of DNA sequencing. Because while the Human Genome Project was wildly successful in generating the first sequence of the human genome, it also came at a pretty high price. In fact, it cost about $1 billion to generate that first human sequence as part of the Human Genome Project. And we realized if this was ever going to have an impact on understanding human disease and eventually be used as a clinical tool, we would need to reduce the cost of sequencing the human genome substantially. And it would have to be something like 6 zeros coming off of that figure. And that's actually in 2003, the notion of the $1,000 human genome sequence was put out as an overt goal. Recognizing that the factories that were used for sequencing that first human genome sequence as part of the Human Genome Project would need to be replaced by newer technologies that would be substantially cheaper than the cost of sequencing the human genome as done in the Human Genome Project. The good news is fast forward about another 10 or 15 years, and we achieved that through a number of new technological innovations and all new methods for sequencing DNA. We can now sequence the human genome for less than $1,000, and the price continues to drop every single year. And what that set up, which becomes very important for rare disease research, was the ability to not only sequence one genome by the Human Genome Project, but to now go off and study tens of thousands, hundreds of thousands, indeed millions of human genomes, and begin to catalog human genomic variation. So across our 3 billion letters of the human genome, any two of us differ about 1 out of 1,000 positions. We're 99.9% identical, but we're 0.1% different, which means that any two of us have at least 3 to 5 million places where our DNA spelling is different. And there's great interest in cataloging, and has been great interest in cataloging that variation and eventually figuring out which of that variation is functionally important and which of that is medically relevant. In order to figure out what's functionally important, you need to understand a lot about human genomic variation. And in chapter 2, we greatly advanced our understanding of where are the functional sequences in the human genome, where are the genes, where are the parts of DNA that get made into RNA, where are different elements that regulate when genes get turned on, and so forth. And over again, we've seen better and better studies come out that help us get sort of Google Map-like views of the human genome, such as that's shown here, that you could access on the internet through any number of browsers, that give you ideas about where are the functional sequences in the human genome, such that when you see a variant sit down in one of those sequences, we'll be poised to be able to try to figure out if that variant is influencing genome function, biological function, and maybe having some relevance to human health and disease. Well, by around 2010, which is now about 12 years ago, progress in human genomics was really quite impressive, so much so that it was time to more aggressively pursue applications of genomics to medicine, inching us closer to the realization of genomic medicine. And that's what Chapter 3 became all about. It was basically putting us en route to genomic medicine implementation. And here the big question was, can we demonstrate effective uses of genomics in medicine? What was that going to look like? And once again, we went through a strategic planning process, and in 2011 published a strategic vision that overtly called out the need to move us towards genomic medicine as a key part of the maturation of the field of human genomics. And this strategic vision would guide us through the rest of the decade. And key to this part of the genomic story was, as I was just alluding to, figuring out which of the subset of the millions and millions of variants that exist, different spellings in human DNA across the world population, actually influence our traits. And particularly with respect to human disease, among those traits, we're most interested in those that are relevant to human health. And here is exactly where the story of rare disease research greatly accelerated. So let me give you a number, and then let me point something out to you. What I didn't mention, but want to mention now, is that the day the Human Genome Project began in 1990, the day the project began 32 years ago, there were 61 rare diseases for which we knew what the mutation was, what was broken in the genome that caused that disease. One of the examples was sickle cell. 61. And of course, I think as all of you realize, there's something like 7,000 to 8,000 rare diseases that we now know that exist, but there were only 61 that we knew the genomic basis. We knew what gene was broken in the human genome to cause that. Well, we knew that this was going to be a very important part of the story of genomics, because we recognize that all of human disease has some elements of genomics playing an influence, but rare diseases in particular are simpler at a genomic level, such as represented here. Rare diseases, also known as simple genetic diseases or monogenic, one gene, or Mendelian disorders. These are all synonymous words, because these are single gene disorders where there's one gene that is broken in the genome that is the major cause for the disease. And while there might be other influences, such as environmental factors or other genomic variants might influence it, the bottom line is this pie chart is the case for about 7,000 to 8,000 known rare diseases. So I told you the number is 61. Guess where we are now? Today, because of advances in our ability to inexpensively sequence human genomes, understand and catalog genomic variants, and figure out how genomic variants influence genome function, we've now been able to characterize and determine the gene that is mutated in nearly 6,000 rare diseases. So we went from 61 32 years ago, now it's 6,000. And yes, we have another couple thousand we need to figure out, but this has been one of the greatest triumphs of human genomics research, that is rare diseases. Now, contrast that to more common genetic diseases. These are also known as complex diseases or multigenic, which are much more difficult to study. And of course, there's thousands and thousands of those, but they don't involve a single variant, a single mutation, a single gene, and they often have a much greater contribution and interplay with the environment. And there's been great progress there, but I just want to contrast to you how much more the substantial progress has been made for rare disease research compared to more common disease research, because the genomic architecture is simpler with rare diseases compared to common diseases. Well, by the middle of the last decade, things had really started to settle into place with respect to efforts to apply genomics in medicine. We had cheap genome sequencing, catalogs of genomic variants, functional understanding of the human genome, progress in disease gene identification, particularly for rare diseases. And these developments really brought us to a position where we started to see genomic medicine come into focus. We started to see genomic medicine truly be implemented, albeit just on limited situations and in only certain clinical aspects. And it was just the tip of the iceberg, but absolutely genomic medicine implementation was here. And let me just briefly tell you the specific examples that I always like to cite. The second one will relate to rare diseases. The first one is cancer genomics, which is not related to this workshop, but the bottom line is cancer is a disease of the genome, and there's been major advances now seen in the use of genomics for the clinical management, diagnosis, even treatment developments associated with cancer. And cancer care is completely getting changed because of genomics. What this workshop is all about is rare disease genetic diagnostics. And here, of course, it has just been game-changing because of the growing knowledge of the genomic basis of rare diseases, coupled with the power of inexpensive genome sequencing, then becoming a diagnostic modality. You are going to hear this afternoon from Bill Gall, who is absolutely a world expert in this, and he will talk about his development of a program first at NIH and then went nationally in the United States and even now extends around the world, dealing with undiagnosed conditions. All of these are rare diseases. My institute has been instrumental in trying to capitalize and catalyze advances in rare disease research using genomic approaches, in addition to the Undiagnosed Diseases Network, which we've co-led on behalf of all of NIH. Our institute has been involved in a program previously known as the Centers of Mendelian Genomics. When it was renewed, it's now known as the Gregor Consortium, which has really aimed to figure out the genomic basis for the rare diseases for which the genomic basis has yet to be defined. And we are getting further and further reaching progress towards seeing a comprehensive knowledge of the genomic basis of rare diseases. And as you'll also hear about later, I even think today, we were involved in a major program that first put the idea of rapid genome sequencing on the map for acutely ill newborns, recognizing that there's incredible opportunity to use genomics diagnostics for helping us in a situation where you have a newborn acutely ill with an unknown recognition of what is wrong with that child. So you're going to hear more about these programs from subsequent speakers. Other things I often like to cite in terms of genomic medicine implementation include prenatal genomic testing, especially non-invasive methods that use maternal blood, and using the new methods of DNA sequencing allow you to screen for chromosomal abnormalities by identifying the small amounts of fetal DNA that naturally float around in the maternal bloodstream. And in fact, it's estimated that worldwide, something like 5 to 7 million pregnant women a year are now getting this non-invasive genetic testing, prenatal genetic testing done. There's also a lot of excitement around a growing set of medications for which genomic information can help guide which among a list of medications is the best match for that patient. That is a practice known as pharmacogenomics, pharmacology and genomics, simply recognizing that each of us respond differently to medication, and a lot of those differences are reflected by spelling differences in our DNA. If we only knew what the spelling differences were in advance, we could pick the best medication for us, for each person, based on the available set of medications. And again, not relevant so much for rare disease diagnostics, but more relevant for common disease diagnostics. There is some growing interest around polygenic risk scores, which can be used to predict a patient's risk for common diseases. This is an active area of research. I would not claim this is being implemented in genomic medicine, but there are major studies that are now looking to see whether polygenic risk scores can be helpful in the practice of medicine. So stay tuned for that. But again, it's not so much about rare diseases. So when we really look at where we were as we were ending last decade, we were celebrating a lot of excitement around the first 3 decades of genomics. We had sequenced the first human genome as part of the Human Genome Project. Crossing that off our list, we reduced the cost of sequencing a human genome by more than 1 million-fold. We went on and sequenced millions of human genomes, cataloging hundreds of millions of different genomic variants across the world's populations. And we were increasingly advancing our understanding of how the human genome works and using all of those tools to significantly advance unraveling the genomic basis of human disease, particularly rare diseases from 61 to nearly 6,000. And as I just pointed out on the previous slide, we had vivid examples of genomic medicine now emerging, including improving the diagnostics using genomic methods of rare diseases. So there was a lot to celebrate as we embarked on this new decade, and we recognized that this new decade really needed to see a new vision for genomics. And so we created that vision, one that was focused on making genomics mainstream in medicine. And the big question we have that we're currently operating on in Chapter 4 is, can we deploy genomics broadly and also equitably in medicine? Can we build on that list of genomic medicine implementation areas? And can we do it in a fashion that's equitable and fair across the world? And we've now articulated this in our latest strategic vision, which was published in October of 2020, which lays out a vision. I hope, and again, if you haven't read this, I would strongly urge you to go to this website and learn about the strategic vision and download a PDF where you will see in 10 pages written in Nature a very clear articulation of the kinds of goals that we have, many of which relate to what will be happening in rare disease research in the coming decade. And at the end of the day, as you will see if you will read this, our strategic vision, there's 4 major areas that we describe. One related to enhancing and adding on some guiding principles and values that are associated with all of genomics. Some of it relates to building a foundation with all researchers can capitalize on advances of technologies and data resources and other aspects of genomics that serve as a foundation. And all of rare disease research is really built on a foundation of genomics. We also have a section where we talk about breaking down barriers, among them being more high throughput and our ability to understand how a genomic variant influences genome function, if indeed it does influence genome function. And newer methods are needed to make that more efficient. When those methods come to be, it will be greatly beneficial to rare disease research. And finally, we describe some compelling genomics research projects. Among them is getting a complete understanding of the architecture of the remaining few thousand or couple thousand of rare diseases for which we don't yet know the genomic cause. And all of these things together are sort of the exciting things that are now being pursued at the forefront of genomics, which is now our Institute's organizational mantra. And so in the interest of time, I was going to talk a little bit more about some bold predictions, but I will leave that to you to simply read and in the strategic vision. And I think I'm going to stop there because I want to make sure I gave you an opportunity to ask questions. So I'm going to stop sharing my screen and turn it back over to the moderators to tell me what kind of questions the participants have.
Dr. Green, we're very tempted to ask you if the bold predictions are quick, to just go ahead and do them. We need to hear from you, and when we need.
Well, I actually have it's about a 3-minute video. Do you want to hear it?
Yes, show us the 3-minute video.
Okay, all right. I'll show you this video because we made bold predictions at the very end of our strategic vision. Are you seeing this, or do I need to reshare? Oh, wait.
You need to reshare.
I need to reshare. I'm sorry. I took you off, and then, okay, let me do this. And then I'm going to, okay.
Genomics, which is the study of all DNA in an organism called the genome, is changing the way we practice medicine, how we manage and treat some previously incurable diseases, and our relationship with society at large. Since the 1950s, double helix and DNA have become household words, standing in for the historic discovery of the 4 chemicals that give us life, commonly abbreviated as A, C, G, and T. Genomics is at an age of innovation and invention like no other, all thanks to how strings of those 4 letters encode the information for creating and operating something as complicated as a human body. Springing forward from the success of the Human Genome Project, a massive reduction in the cost of sequencing a human genome, and the dawn of the CRISPR gene editing era, genomics is leading into an even more audacious future, a future that will define our collective lives for decades to come. But what could this future look like? Here is a summary of 10 bold and fantastical predictions made by experts at the National Human Genome Research Institute and the genomics community for the next decade of human genomics. While most are unlikely to be fully attained by 2030, achieving even one or more of these by that time would have lasting impact for science, medicine, and society. Prediction 1. Sequencing and analyzing a complete human genome will become commonplace for any research laboratory. Prediction 2. The role of every gene in the human genome will be known. Prediction 3. Environmental influences on our genome will be routinely used for making predictions about health and disease. Prediction 4. Genomics will no longer use social constructs such as race in human research studies. Prediction 5, student projects involving the study of millions of people's genome sequences will be regularly featured at school science fairs. Prediction 6, genomic testing will become commonplace in medicine, just like a standard blood test is used now. Prediction 7, it will be readily known if a given letter difference in a person's DNA is clinically important. Prediction 8, a person's complete genome sequence will be available on their smartphone in a user-friendly form. Prediction 9, advances from human genomics will benefit all of society and not just a few. Prediction 10, genomic discoveries and technologies will help cure more genetic diseases than ever before. How do we get there? The National Human Genome Research Institute is committed to leading research to make such predictions a reality. Learn more about the Institute's 2020 strategic vision for the future of human genomics by visiting genome.gov/2020SV. Dive deeply into a field that continues to transform medicine and will hopefully continue to bring curiosity, purpose, and compassion for people everywhere.
So Rachel, it was a good place to stop because it brings us back to the very first question you asked me, which was sort of, are we at an inflection point? And while those bold predictions are very audacious and they were deliberately meant to point to the future, not being sure we'll make them in 10 years, but would it be great if we did? I counted as I watched the video, which I've probably seen 100 times, I counted at least 4 of them are directly relevant to rare diseases. And if we make progress in moving towards those goals, it will bring the kind of inflection point that you asked about at the very beginning with respect to rare disease diagnostics, understanding of the disease, and potentially treatments, as the prediction number 10 talked about.
I'm so glad we were able to see that film together as a group, because I think the impact of these predictions is absolutely breathtaking, stunning to me. And I'm so grateful that we were able to work through these technical difficulties and that you were able to stay. We're going to figure out a way to corral you back in to talk to these journalists about this issue. And I want you to know that they are putting questions in the chat. So we are going to compile these questions and try to get them answered for the fellows by the end of the workshop, or the training rather. But Dr. Eric Green, thank you so much for your time. And again, we will be in touch with you.
Okay, it's great being with you today. Take care.